of genome editing tools and their translational benefits today. I hope this topic has not been covered in the previous talks. Nobody has covered this, but this is something that you would like to learn, and you must be hearing a lot about this genome editing thing, right? So when we talk about genome editing, what comes to your mind? What are the different techniques that are available? Zinc finger nucleases, right? Talons are there. Then CRISPR-Cas. CRISPR-Cas has revolutionized the genome editing, field altogether because of its, various benefits that it offers. We are going to look into all this. Okay, so today's topic is basic understanding of genome editing tools and their translational benefits. But before the advent of this genome editing thing, before even the advent of CRISPR-Cas9, CRISPR-Cas9 has been recently introduced, right? In 2012 only, if you know, who was given the Nobel Prize for, in the field of chemistry. 2 scientists received Nobel Prize for CRISPR-Cas9 application for genome editing. Jennifer Doudna and Emmanuel Charpentier. Okay, these are the 2 ladies who received the Nobel Prize. So it was a big discovery for which their contribution was recognized by this Royal Swedish Academy of Sciences. So what questions come to our mind? So we'll start with what is genome engineering? Genome editing or genome engineering? You keep on switching these words and use them interchangeably. So genome editing or engineering is, what do you understand by genome editing engineering? So genome of any organism is kind of fixed, By random mutagenesis only it can be altered. But if you want to specifically alter the sequence of any stretch of DNA, then you need to have some editing tool that will precisely edit that part of the DNA. For that, you need to have certain tools. You cannot use random mutagenesis in that case, because random mutagenesis, you will not have a handle over which sequence is going to be mutated. Because of that reason, you need to have some gene editing tools. So genome engineering is actually manipulating the sequence of the microorganism or plant or animal so that you will have your precise DNA sequence in the engineered organism. Okay, whether it be a single nucleotide substitution, addition, deletion, mutation, any kind of mutation that you can think of. So that is genome engineering. Then what are its applications? We are going to see what are its applications. So this genome engineering has been used for various applications now. You name it and it is being used for that kind of application. Whether it is in crop improvement, human health, increasing the benefits of the microorganisms for this human use, whatever you name it, even for gene therapy purposes, it has started being used. How it can be performed, that also we'll learn. So basically, we are going to focus on 3 different tools. One is, as you said, zinc finger nucleases. The other one is talons, and the third one is the most predominantly used nowadays because of its usability to the user, ease to use. It's CRISPR-Cas9 genome editing. Okay. But before going into genome editing tools, what I was referring to is the random mutagenesis. So before you go into the precise genome editing part, you should understand that you can even use random mutagenesis to elucidate the function of any target gene. Suppose you want to elucidate the function of X gene in microorganism. Okay, in bacteria, you are interested in identifying the function of certain gene. Okay, so then you will use certain specific tools to identify the function. But if you want to just use this tool to introduce random mutagenesis and ask the function, like this function has been acquired because of what change in the genome, then you use these random mutagenesis. You make a library of mutations in the microorganism, select for them, and then see have they acquired any new function or not. That you do by just random mutagenesis. So random mutagenesis is the process whereby you just randomly mutate the genome without having precise control over which stretch of the DNA will be mutated. That is called random mutagenesis. Okay, so for that you need to have a mutagen. Mutagen is any substance that would assist you in mutating the genome of an organism. So like mutagens can be in different forms, like chemical mutagens you must have used, you must have come across in your, so what is the background of some of, you can tell like from which background do you come? BSc, MSc? You all are masters? So, okay, so but you have basic understanding of the microbiology and the genome engineering part, little bit of it? Okay. You have done this recombinant DNA technology coursework? Okay, so you would understand this. So I hope you would have come across these chemical mutagens like EMS, ethyl methane sulfonate, and ethyl nitrosourea, all these. So these are the chemical mutagens that are used as alkylating agents to precisely, make substitutions in the DNA sequences. So like for instance, this ethyl methane sulfonate, EMS, is predominantly used to introduce this substitution from, this is used for transitions. Any idea what this EMS does? It changes GC to AT, okay, because it alkylates this O6 position in the guanine, so that it mimics A but then pairs with T. Okay, it is supposed to pair with C, but after treating with EMS, what happens is this O6 position is ethylated. That causes it to then pair with T instead of C. So in the next round of replication, what would happen is that GC will change to AT. Okay, likewise, this N-ethyl nitrosourea is also used for the same purpose as an ethylating agent, and it is called supermutagen because it can also lead to conversion, transition of not only GC to AT but AT to GC also. Okay, so these are very powerful reagents to perform this random mutagenesis. And what is the benefit of random mutagenesis? That you use random mutagenesis to identify new functions in the microorganism. You treat your organism with these agents, make a library, and then see what new function they must have acquired. Now, how would you identify what is the gene responsible for this mutation? See, you have just mutated the microorganism, but you do not have any idea like where this mutation has been incorporated that is leading to the functional change. So there are two things. One is changing in the sequence and leading to the functional change, that is the phenotype. Yes, so very nice. So what is forward genetics and what is reverse genetics? So when I randomly mutate the genome of any microorganism and then look for the phenotype and go back and try to identify which gene is responsible for that, is it forward genetics or reverse genetics? So you understood what I'm asking? We have incorporated the mutation, we have a phenotype, now we want to trace back what is this phenotype due to. This phenotype is due to some gene, some changes in the DNA sequences. That is reverse genetics? All of you? And if I know the gene, I manipulate that gene and identify the function, that is? No. It is the other way around. Okay, so forward genetics is when you have the phenotype and you trace back the gene. That is forward genetics. And when you know the gene, you try to identify the function, that is reverse genetics. Okay, so just remember this. So in addition to these chemical mutagens, there are other agents that are available for random mutagenesis purposes, like for instance, physical means like X-rays, UV radiations and all. Those can be used. And I have written this random mutagenesis via EMS is a powerful agent that is being predominantly used for random mutagenesis purposes to identify what the new function has been acquired by the microorganism and tracing back it to the gene level, polymerase chain reaction. So polymerase chain reaction, the way it works, you need to incorporate the primers and you have to have a DNA polymerase, then that will amplify the target gene sequence with the primers. So can that PCR be used for random mutagenesis? Site-directed mutagenesis, very nice. So in the site-directed mutagenesis, how do you incorporate mutations? Primer sequences you change. But can PCR be used for random mutagenesis? Because we are talking about random mutagenesis. Site-directed mutagenesis, you are very right, that you can incorporate these indels, you can delete, edit, and you can even make substitutions using these changes in the primer sequences that would allow you to precisely regulate where you incorporate the mutation through PCR. But for random mutagenesis, yes, PCR can be used. You must have heard about one PCR, that is error-prone PCR. You have heard about error-prone PCR? So what happens in that? You specifically use the PCR mechanism to randomly mutate the amplified sequence. The way it is done is, so you use magnesium, right? Magnesium MgCl2 is used as an agent that would be utilized by the DNA polymerase, okay, to stabilize this, base pairing between the sequences. But if instead of magnesium, if you use manganese, that would allow these improper pairing to be incorporated. Even changing the concentration of the magnesium itself will allow you to increase the mutagenic frequency or the frequency of the incorporation of mutations in the PCR. That would cause it to become error prone. That is why it is called error prone PCR. Okay, and in addition to that, the infidelity of the polymerase itself. Like if you use a polymerase, now there are a lot of different versions of Taq polymerase that are available that allow you to even correct the incorporated mutation. But if you intentionally use a polymerase that has high frequency of introducing mutations, mutazyme, they are called mutazyme, then you would increase the error prone rate. Okay, by all these means, you can increase the random mutagenesis through PCR. So PCR is not only used to amplify the target sequence, it is used for various different purposes. One of them is random mutagenesis. Okay, any question? No? Okay. So since we introduced this, we are going to touch a little bit more on this, how to increase this. We have seen about random mutagenesis. So gradually we are going to shift and see how now from random mutagenesis to precise mutation through in vitro means to editing of the genome will move like that. Okay, so now we have seen random mutagenesis through these mutations, PCR. Now the use of the PCR for incorporating specific changes in the amplified DNA. Like some of you said, site-directed mutagenesis. So site-directed mutagenesis can be used to incorporate specific mutations in the target amplified DNA. How it is done? Like for instance, in this example, you want to add something to the 5' end of the gene that you are interested in amplifying. Then how will you do? Using PCR. What will you do in your primers?
प्राइमर में दोनों साइड थोड़ा सा कॉम्प्लीमेंट्री सीक्वेंस रख के
Right.
इसके एरिया में अपना डिजायर्ड सीक्वेंस डालेंगे जो हम
Very nice. So in the forward primer, you are going to add the thing, the nucleotide sequence that you are interested in adding to the amplified DNA, but having the complementary sequencing flanking that additional sequence. So as if you see here, so this is the forward primer, right? This is the reverse primer. So what you have done in the forward primer is that these are the complementary sequences, and flanking this, the sequence that you want to add, you have these complementary sequences. So you need to have complementary sequences flanking the sequence that you want to add. in the forward primer, not in the reverse primer. You can do the same thing in the reverse primer if you want to append something at the 3' end. Clear? So what will happen in this case is, this is going to amplify like this, you will have a gene product. Then as the cycles will keep on increasing, you will have predominantly more population of this DNA which has the addition. Clear? Okay. Now, if you want, as I was describing you earlier, so if you want to append something at the 3' end of the gene, then what you do? You do not change the forward primer. What you do is, you change the reverse primer. In reverse primer, at the very end, you put something, the additional sequence, not in between. Okay? So you have added here this. Clear? Now if you want to delete something, then what will you do? Suppose you want to delete this region in the target sequence. You don't want that sequence. I am interested in studying what will be the function of deletion of this particular nucleotide stretch on the functionality of the gene. So what will I do? I need to delete this region. How will I delete that? Same way. Like in the previous example, what I showed was, you have something that was looping out, okay, in the primer. But here, that something will loop out in the target sequence, because that doesn't have a complementary sequence in the primer. So if you want to delete this, you will design your primer in such a way that you do not have that target sequence in the primer, in the forward primer. Making sense? Any question? Okay, so now you have this, then you will have a final product. As the cycle will keep on increasing, it will have predominant population of the amplified product that has the deleted segment of the gene. Clear? Okay, now if you want to add something, primer extension mutagenesis, if you want to add, what we saw was addition of a sequence at the 3' end of the gene. That was very straightforward, because you just need to append something to the reverse primer and then it will be incorporated into the final amplified product. But what if you want to add something right in the middle of the gene? How will you do that? If you want to add something right in the middle of the gene, so this is the target sequence, but what you want to do is, you want to add something right in the middle of the gene or somewhere in the center of the gene. Then how will you do that?
So if we synthesis primer which is inverted of this template sequence.
Not inverted, no. You need to always have this sequence. In one case, you will have that sequence in the reverse primer, and in the second segment of that PCR, you will have that sequence in the forward primer of the second PCR. So just focus on this. So you want to append something right here, right, in the middle of the gene. So what will you do is, you will not touch the forward primer of this. This will be as such, but in the reverse primer, you will have some overhang. That overhang will be essentially the part that you want to insert in the DNA. Okay, and in this second one, second PCR, you will have the same overhang, complementary sequence of the same overhang in the forward primer. So this and this will be complementary to each other. Okay, so in this PCR, what will happen? You will have one fragment AB, and AB, when you will get this product, so you will have A, B, and B will have a tail. That is one PCR. In the second one, C and D, you will have C, but at the 5' end of the CD amplicon, you will have C as a overhang. On the C, you will have an overhang which you want to append. So now, in the next cycle, what will happen? This will provide a complementary phase. They will complement with each other. Okay. Likewise, you can insert any length of sequence in your primer, in your amplified product. Clear? Any question?
So
Last part, this one. Okay.
Segmented joints.
Yeah, so maybe I can use this, right? This is A, this is B. Okay. At AB, you will have, at the end of the B, you will have this additional part. Okay. Now with CD, you will have at the starting of the C, you will have that additional part you want to incorporate. Now in the second step of the PCR, what will happen is, so you can do in a one-step PCR the same thing, or you can have two different PCRs. You do PCR, you have the same target DNA, do AB PCR separately, CD PCR separately, purify them or take these as a template and then mix them together. But in that round of replication, you will have only A and D as a primer. Because that complementary sequence, the binding sequence of both the templates is being provided by this complementary region. Or it can be done in the same single PCR step also. So if you do them separately, what will happen? Just consider this, these two. AB and CD you have mixed together. Now what happens here? This is 5', 3', right? So what will the polymerase do? The polymerase is going to extend this. Similarly, the polymerase is going to extend this. So you will have a complete sequence. Understood? First the polymerase is going to extend this. Clear? So see, like this. This is the DNA sequence. Okay, you want to add something here. So what you have done is, you have used this. In the reverse one, you have added something. Right? And similarly, you have used this primer, but you have added something here. And you have a reverse. Reverse you do not touch. Forward of this and reverse of this you do not touch. Only the reverse of the first one and the forward of the second one you have to have a sequence which you want to add. Then in the next step what will happen? You will have this where you have this sequence. Okay, and in the next one you will have like this. So what is providing the complementarity for joining of these two amplicons together? The region that you want to incorporate. That is providing the complementarity. So the polymerase is going to extend this and this first. And then since you have the primer pair A and D together, they will just like act as a normal PCR. Understood? No? So just go back and do it yourself, then you will understand it better. Okay, these are two separate PCR. Just consider them as two separate PCR, mixing the products together and re-amplifying using the extreme primers, A and D. Okay. Okay, similarly you can delete regions. You do not incorporate this. Okay, if you want to delete something right in the middle of the gene, you will not incorporate that particular segment in the reverse primer of the first PCR. Similarly, you will not incorporate that sequence in the forward primer of the second PCR. Like, see, focus here. So A and B. In B, you have this particular sequence complementary to the region just upstream of the region that you want to delete, and the next region that is here is actually complementary to this. So the region which you want to delete is not at all represented in the reverse primer. Clear? Similarly, in the forward primer of the second one, CD, you will have a region which is flanking, which is corresponding or complementary to the region upstream of the deleted region in the forward primer. And then reverse primer you do not touch. So what you end up getting is the first amplicon as the one which do not contain this particular deleted region. Then you amplify the whole thing with A and D. That's how you can delete region, you can put in regions. Okay, clear? Okay, so now what we have understood so far is how to do random mutagenesis. Just randomly mutate the genome, identify the function, and then tracing it back to what might have caused this particular change in the phenotype. To something that we can precisely control under in vitro conditions to understand the function of the gene. Like if I want to understand what will be the effect of deleting a particular residue on the activity of some enzyme. So what I'll do is I will make the amplified gene in such a way that I will not have that particular residue in the amplified product. And at the same time, I will also make a particular gene which is having the intact sequence so that I can compare. So intact का ये function है and then deleted का what is the change in the function. So that will allow you to identify the function of the gene. Okay, clear? So now if we want to do the same thing precisely in the genome, that's where the utility of these genome editing tools come. If I want to precisely ask a question, now I have shown under in vitro conditions that deleting this particular nucleotide from X gene is going to affect its function. Okay, now I want to understand what would be the repercussion of this particular substitution or deletion on the phenotype, on the overall phenotype of the organism, because I have just been able to elucidate what is the functional consequence on the gene function. But if I want to understand what will be the functional consequence on the phenotype of an organism, then how will I understand that? I have to introduce this into the genome of the organism and then ask a question, now what is the effect of this gene, mutated gene, on the overall function of the organism? Clear? So that's where the utility of the genome editing tools come from. Clear? So this is how it has been shown in this cartonic representation that you are precisely substituting a single nucleotide from millions of bases. Just imagine you have millions of bases in the genome. Okay, you want to precisely take out one and substitute with another, or you want to precisely delete a single nucleotide, let's say, from millions of bases. It looks really a mammoth task, complicated. How will you do that? But there are tools available. As we said that there are tools available starting from zinc finger nucleases. Even before the advent of the zinc finger nucleases, there were these enzymes, just like the restriction enzymes work. So how do restriction enzymes work? Restriction enzymes actually recognize a particular stretch of sequence. When they recognize this sequence, they sit on that and then they make cut at the designated, between the two designated nucleotides. Okay, but the problem, we cannot use these restriction enzymes everywhere for genome editing is because
it can cut in multiple sites.
that is one thing. Another one is the gene that you desire to manipulate may not have site for restriction site. Right? So that's why you cannot use them. You have to use something which you can adapt to your use. Okay, so one of the important functions of any gene editing tool is its adaptability to the user. Whether the user can adapt it for the precise genome editing of the target gene that he or she might be interested in. Okay, and another important feature of any gene editing tool is that it should be specific. Suppose you want to edit something but you end up editing something else. Do you want to do that? No. So it should be precise in its function. There should be very less off-target effects. You must have heard this word regularly in, I don't know how many of you follow this, but in these gene editing tools, one of the problems is off-target effects. You want to do something, but unintentionally or inadvertently, you would end up also mutating something else that you don't want to do. Okay, so two basic requirements of any gene editing tools is that it should be precisely used to manipulate your target gene, and also it must be adaptable, must have sufficient adaptability for retargeting of any user-defined sequence. Today you want to manipulate gene X, Tomorrow you may want to manipulate gene Y, A, B, C, D, and so on and so forth. So you should be able to adapt that tool to your use. Clear? So this is where the function of these gene editing tools come. So we were discussing about these restriction endonucleases. They cannot be used as precise gene editing tools. There is one more tool that is available that is called meganucleases. How many of you have heard about meganucleases? So that is also one of the tools. But again, the problem is meganucleases were discovered initially as gene editing tools. They recognize a very big sequence of nucleotides, around 35 to 40 nucleotides. But the problem is again that those sequences are very rare. Okay, they may not be present in the segment of the gene that you want to manipulate. Okay, and it's very difficult to adapt them for the user's requirement. So that's why they were not developed so much. But the gene editing tools that really gained attraction starts from the zinc finger nucleases. So zinc finger nucleases consist of, so in any of these gene editing tools, you will have two components. One is for precisely recognizing the target sequence, and the other component would be the one that is going to make the double strand break. See, you want to first take that system to the target site. Okay, I have that particular stretch of DNA where I want to take this particular tool. I would take my enzyme there, but the precise cut is going to be incorporated by the endonuclease. That is in conjunction with the recognition sequence, the recognition part itself. So there are two components. One that is going to recognize the target sequence, the other one that is going to introduce the double strand break. That's the only function of gene editing tool, to take that particular endonuclease to the target site. Once that endonuclease has been taken to the target site, it will introduce the double strand break. Then now it is the onus of the repair machinery of the cell to repair that. So once the double strand break has been introduced in the target genome or anywhere in the genome, what is the next step that the cell would do? To repair that, because it is detrimental for its survival. Because you cannot have a cut DNA chromosome and then the cell is going to die. So the next thing after the introduction of the double strand break is to repair that double strand break. That can be done in two different ways. One is homologous end joining, non-homologous end joining, and the other one is homology directed repair. Okay, so non-homologous end joining, NHEJ pathway, that is error prone. Okay, the other one that you can use to your advantage to precisely manipulate the gene is the homology directed repair. So you yourself provide a repair template. You ask the cell to use that repair template to repair that particular double strand break. And during that process itself, it incorporates whatever you want to desire, whatever change you desire to incorporate.
Sir, in vivo condition, we can promote homologous directed mutation repair.
In what?
In vivo condition.
In vivo condition, okay.
But it is too time taking. So can we use NHEG?
It is too time taking.
Compared to NHEG?
No, it's not like that.
Not like that.
It's not like that. It's not time taking. So if your aim is to just random incorporation of the DNA sequence, NHEJ, you do not have a control.
So in many cases, why we prefer NHEG?
In what conditions? Okay, so if your aim is to just disrupt the sequence and ask the question what would be the change in the functionality of the gene, it's just like random mutagenesis. Okay, you want to disrupt that particular sequence of target gene, you just target it, introduce the double strand break, and then there will be frame shift mutations during that repair, addition, deletion, indel happens. So that would lead to loss of function of the gene. Then you ask the question, what is the consequence of this loss of function of gene? In that case, you do not provide a homology or the repair template. If you want to precisely ask a question like what would be the change if I introduce this mutation or this substitution, what would be the change, then you do not depend on NHEJ. You have to use the homology directed repair. Okay, clear? Okay. So now, in any of the gene editing tools, you will always have these two components. What are those components that I just described? One is the recognition sequence, or not the sequence, the recognition component. What component of this gene editing tool is going to take my endonuclease to the target site? That is one component. The other component is the endonuclease itself. Because what you want to do is, you want to take that particular endonuclease to the target site. That's the only function of gene editing tool. Okay. So in this zinc finger, the thing that actually recognizes the target sequence is the zinc finger protein. So you have these zinc finger proteins that are placed together in tandem that recognizes the coding sequence and the non-coding part of the DNA. So you have this, the left zinc finger protein and the right zinc finger protein. So they recognize two different parts of the DNA, the target sequence. This is called the left zinc finger protein, this is called the right zinc finger protein. And at the end of these, you have the endonuclease that is called FOC1. So the FOC1 is only going to work when it combines with another FOC1. So by itself, it will not just, so if you have only the left zinc finger protein introduced into the system, into the cell, it's not going to work. Only when they are going to come together, when they recognizes a target sequence, so this component recognizes this component recognizes this, and this is reconstituted. The FOC1 endonuclease is reconstituted. Then it makes a double strand break. Clear? So this zinc finger protein, as such, you must have heard about zinc finger proteins. So these are small proteins that have particular motif. There are different flavors of zinc finger protein that you see. The most prominent one that is used for this particular editing thing is the C2H2. 2-cysteine, 2-histidine, and then there is a zinc that coordinates with 2-cysteine and 2-histidine. Okay, this will be clear here. So this motif actually recognizes the DNA sequence, but only 3 nucleotides. So this alpha helix that you see, so there is an alpha helix in this zinc finger motif, and you have an antiparallel beta sheet. So this is the recognition helix. This actually recognizes the base pairs. So then you have combination of these zinc fingers. So this single zinc finger motif actually recognizes 3 nucleotides. So now in this example, how many nucleotides this is going to recognize? This particular zinc finger protein combination of 4 tandem zinc fingers is going to recognize how many nucleotides? 12. 4 3s are 12. Simple. Okay. So to increase the specificity, to have the the basic requirement is that your gene editing tool should be precise, and that can only come if you recognize a longer stretch of the DNA. So this is 12. So 12 nucleotides together in a particular sequence is very, it's not that frequently found in the genome. So to further increase the specificity of this, you can increase one more zinc finger motif into this. That would recognize 15. 3 more. Okay. So you have zinc finger protein anywhere from 4 to 6 to increase the specificity of the system. In this example, 4 motifs are placed together in tandem to recognize the sequence in the coding DNA and the non-coding DNA. When they come together, then only they will be able to introduce the double strand break through fork 1. Clear? Any question? So here you focus here. So this particular zinc finger motif is going to recognize GGG. This is actually designed in such a way that it is going to recognize GGG. Similarly, you can design zinc finger motif that is going to recognize GGT. Similarly, you can design something which will recognize GAC. Then if you have a stretch of this 9 nucleotides together, you will combine all these motifs together to make a tandem sequence, protein sequence. That's going to recognize this particular stretch of 9 nucleotides. Why it did not gain so much popularity, zinc finger? Because it's not easy to functionalize these particular motifs to recognize a particular stretch of amino acids, these nucleotides. Okay, that is something that is proprietary to some companies. You have to submit your sequence to them. They will design these zinc fingers for you, and then you can use them. That's not user friendly, right? Why would I have to go every time to someone to ask to make something for me when I have an option of making it myself in the form of the better one, CRISPR-Cas9. Making sense, right? So similarly, if you want to edit something or recognize some other sequence, then your sequence may change. So likewise, you will have to change your zinc finger motifs. But the problem is that you will have to depend on the company to make those zinc fingers for you. But now you know how it works, zinc finger nuclease. Okay, so this is the example that I already talked about. So similarly, you see in this ZIF268 protein, you have these 3 zinc finger motifs together that recognizes a particular sequence. So zinc finger proteins generally are used as the transcription factors. In the transcription factors, you have these zinc finger motifs that are used for precisely recognizing a target sequence. That has been exploited to use as a gene editing tool, because you need to have a recognition part. You need to have something that will recognize the target sequence. So these double strand breaks that we introduced through these zinc finger proteins. So what will be the outcome? The outcome will be either the NHEJ pathway, okay, through NHEJ pathway, as you were asking this question. So you will have this gene disruption that inadvertently it will be introduced, because this is error prone NHEJ pathway. So you will end up either deleting something or adding something on which you will not have a control. So you will end up disrupting the function of that gene. Okay. Largely it is used for that purpose. But if you want to precisely add something, manipulate something, then you would not use this arm. You will provide a repair template that will have your particular change in the repair template. Suppose you want to change ATG to AAT or something. Then in the repair template, you will provide that. Clear? That repair template will be used by the repair machinery to make that substitutions. That will lead into the correction of the mutation. Or you can add something, targeted addition. If you want to add some sequence to the end of the gene, to the middle of the gene, you can introduce that in the repair template. Similarly, you can do this transgene stacking. You want to stack different fragments of different genes, so you will be able to do that. But the thing is that those have to be provided in the repair template. Clear? So these are different functionalities. This is how the double strand break is actually getting repaired, through NHEJ pathway or through this HDR, homology directed repair. Then zinc finger nuclease is still being used, okay, and it has been used for various different purposes, like for instance correcting for gene disruptions, for gene correction, for gene addition, all those sorts of things have been done with and in various different kind of organisms. So one of these examples that you see here is in the gene disruption is this human T cells, you see, for the correction or for this taking care of this HIV infection, because that virus uses a receptor called CCR5. So if you disrupt CCR5, you will not allow the virus to actually interact with the CCR5 receptor. So if there is no CCR5 receptor, it has been deleted through this this zinc finger nucleases, you will correct that particular infection. Likewise, it has been used for other purposes, like for instance this in tobacco gene correction, and in human embryonic stem cells, mouse embryonic stem cells, to make different kind of resistant varieties. For instance, suppose there is a pathogen that infects tobacco, tomato, or any kind of plant. You want to make it resistant towards these pathogenic microorganisms. You know the target gene, so you manipulate the target gene through zinc finger nucleases, and then that infection is not going to be, or that infection is going to be corrected. The transgenic variety will not be infected by these microorganisms. Clear? The next one that we are going to discuss about is talents. Okay. So for how long this will go? 11 o'clock, right? Okay. So we have time. So next is talent. So after zinc finger nucleases, talent. So talent, anybody knows how does talent work? Talent is actually consisting, again, just remember, two components. One is the recognition part, the other one is the endonuclease. Same thing is in talent. So talent has two components. One is tail, the other one is N. Tail is actually a transcription factor that is present in this plant pathogen that is called Xanthomonas. Okay, so that has been utilized, exploited to incorporate or fuse this nucleus to it, because tail is very specific. Okay, so then in talent you have the full form is transcription activator like effector nucleases. So tail is transcription activator like effector, and when you add nucleus to this becomes talent. That can be used for gene editing purposes. So now to understand how the talent actually works, you need to understand the domain architecture of the tail, the transcription activator like effectors. As I said, this tail is actually the transcription, the protein that is present in the pathogenic bacteria, plant pathogen, that is Xanthomonas. So what happens during the infection, this is actually secreted, this transcription factor tail is secreted into the plant cell. Okay, through this type 3 secretion system. It goes to the nucleus, binds to the promoter region of the target genes, and manipulates the gene expression. So what we have done is, we have actually taken this tail and fused it to, again, FOC1 endonuclease. Two components. Okay, so now you understand what is tail, how does it look like. It's a big sequence, big protein sequence. So consists of 3 different regions. One is the N-terminal region, the C-terminal region, and the central region. Central region is the one that is of utility to us. Okay. So if you focus on the N-terminal region has this type 3 secretion signal sequence, T3SS, and the non-canonical repeat. 4 non-canonical repeats join in tandem. Okay, that is just for recognizing any sort of DNA, not very precise. The C-terminal has this transcription factor binding site. It binds to the transcription factor. Like this one, actually tail binds to transcription factor 2A in plants for manipulation of the transcription of the genes. Then you have this NLS, 2 NLS sequences. What are NLS?
Nuclear localization.
Very nice. Nuclear localization signal sequence. So what is the function of NLS? it actually allows any protein which is present in the cytoplasm to the nucleus. Okay, there is a whole machinery for that. Through the nuclear pore it goes. So these NLS actually allow this tail to get imported into the nucleus of the plant cell when it is secreted by the bacteria during infection. Then the other one is acidic activation domain, AAD. Now, so this is one part, this is second part. The third one, which is right in the middle of the gene, is the repeat region. So this repeat region consists of tandem repeats that are highly conserved in sequences. So this is repeat region altogether. There is variable number of repeats. In this repeat region, again there are variable number of repeats. You see these rectangles, red rectangles, tethered together, arranged in tandem. So these can be variable in number, but their sequences are quite conserved, except that there are two highly variable amino acids at 12th and 13th position that are called RVD. Highly variable di amino acids. Okay. So these RVD are actually the ones that are going to give specificity to this repeat region. So one of these repeat regions is shown here, the sequence. These sequence will be conserved here also, here also, here also. But the non-conserved sequence will be these 12 and 13. That will provide specificity to this particular repeat segment to recognize a particular nucleotide. So for instance, if this RVD in a repeat segment consists of NG, this particular repeat segment, repeat region is going to recognize T. Similarly, if this RVD consists of NI instead of NG, then it is going to recognize A. So if you want to recognize, if you want to target a particular stretch of sequence in the gene, and you know the sequence, then you would arrange your repeat region in such a way that you have the corresponding repeats targeting that particular stretch of DNA. Suppose if I want to target TAGCA sequence, let's say 5 nucleotide sequence I want to target. So I will have this repeat region where these repeat segments will be corresponding to NG, NI, NN, HD, NS. I will make my central repeat region in such a way that it will consist of tandem sequences of this. Clear? But you don't target 5 nucleotides, you target a larger fragment to provide specificity. Clear? Any question on this? This is only tail, but at the C terminal, if you fuse the endonuclease, the FOC1 endonuclease, so then you will be able to take this FOC1 endonuclease to the precise site in the genome. So the only purpose of this particular tail will be to target the FOC1 to the target site, like we saw in case of zinc finger nuclease. So zinc finger nuclease was taking the endonuclease FOC1 to the target site for introducing the double strand break, where in between somewhere this FOC1 is going to introduce the double strand break. So here it is more elaborately shown, the central repeat region consisting of different repeat segments. So these RVD sequences only are shown, HD, NN, NG, HD. So why they are shown? Because they are the ones that are going to provide the specificity to this repeat region to recognize a particular DNA sequence. Clear? Like for instance, this one is shown here, HD. So HD is going to recognize C. So this particular repeat is going to recognize C. Only this one. Likewise, NI is going to recognize something else, T. So now if you have this particular sequence, this is my target sequence now. My target sequence is what? CGT, CAG, TTG, CCA. So now I will make the central repeat region in such a way that my RVDs starting from here will recognize C, G, T, C. So this C is recognized by HD. So wherever you see C, you have HD. C, HD, C, HD, C, HD, C, HD. Similarly, if I want to recognize G, I would have NN, G, NN, G, NN. Similarly for AN, G. So you'll make your repeat region in such a way that it will provide specificity to your tail. That is one part of it. The other one you will make against the non-coding sequence of the DNA, both the strands of the DNA, so that it provides more specificity. And also it can only function the fork1 when they are together. When they come together, recognize lower and upper part, then the fork1 is going to introduce the mutation, the double strand break. Clear? So again, various applications of tails. If you want to introduce something at the end of the gene, if you want to modulate the expression of the target gene and you do not want to cut something, okay, that is called dead nucleus. You can take a dead nucleus, you just append something, the modifier, like for instance this methylases, acetylases that you can append on your fork1, and then they will take this particular tail sequences, take your acetylases, methylases to the target site, and those are going to manipulate the expression of the gene without cutting the gene sequence. So you can precisely regulate the expression of the genes by using these tools also. Okay. They can be used, modify the expression of this activator, repressor can be appended to that, genetic modifications like using different base editors that will allow you to convert one base to another base as a base editor. Okay, so now the last one that we are going to discuss is, and the one that you all are familiar with, is the CRISPR-Cas9. So I hope you know how CRISPR-Cas9 works. Anyone who wants me to tell how CRISPR? So CRISPR, can you explain CRISPR?
So sequence will be specified.
Clustered regularly sequence, right? So CRISPR-Cas9 was actually not discovered as a tool for gene editing. Rather, the initial discovery that happened in the CRISPR-Cas9 field is that this particular CRISPR locus is present in bacteria, and it actually acts as an adaptive immune system for the bacteria to be protected against the invading pathogens, invading viruses, invading DNA sequences. To protect itself, the bacteria has actually evolved a system which can restrict the incoming DNA, either from virus or different kind of organisms. Okay, so here what is shown is the Streptococcus thermophilus CRISPR1 locus. So there is a whole locus that is dedicated to this functionality. So there are different Cas proteins. Cas are these endonucleases. They are not only meant to restrict the incoming DNA, they are also utilized for processing the RNA molecules that will in turn be used to recognize the DNA. Okay, so you see this Cas5, Cas9, Cas6, Cas7. So this is how this CRISPR1 locus is actually made or represented in this bacteria, Streptococcus thermophilus. Then there is this region that is called the repeat spacer region. This is the region where the bacteria actually incorporates small fragments of the DNA from the incoming pathogen, so that next time when the same pathogen attacks that bacteria, it already has a memory. So why it is adaptive? Because it has a memory component to it. Why it is called adaptive immune system? Because there is a memory component in form of the spacer sequences that the bacteria puts while during the first infection. So it retains spacer sequences from the invading DNA that it puts in this CRISPR-1 locus. So this is expanded here. You have a leader sequence, then you have repeat and spacer sequences. So these diamond boxes, black boxes that you see, these are the repeat sequences. These are repeat sequences, and between two repeat sequences you have a spacer sequence. That spacer sequence is the one that is going to recognize the invading pathogen. It is going to bind with the DNA sequence of the pathogen. Okay, so this bacteria, Streptococcus thermophilus, was infected with two different kind of viruses, phage, two different phage, 5858 and 52972. During that infection, what was observed was that some of the bacteria have made themselves resistant to infection with these viruses. So now when the scientists traced back what is the reason for this particular bacteria to be resistant to these viruses, what they found was, when they sequenced this particular CRISPR-1 locus, they found that this particular bacteria, which is resistant to these phages, had some additional sequences which do not correspond to the bacterial sequences. In fact, it corresponded to the sequences of the virus. Okay, now then again they showed that this is the wild type sequence. So one of these stretches have been expanded here, leader sequence, repeat, spacer 1, repeat, spacer 2. Now focus on the bacteria that is resistant to one of these phages, let's say 858. So this particular bacteria has this spacer sequence S1 and S2. Now this S1 and S2 are the sequences which correspond to the phage DNA sequence from 5858. When they infected this particular bacteria which contained this S1 and S2 with phage 858, there was marked reduction in the this pathogenicity by this bacteria. So you see this efficiency of plaque formation is, so in the wild type, this particular virus was not able to, the bacteria was not able to grow. There was a high efficiency of formation of the plaque. So this is the efficiency of the plaque. So you see that these bacteria were eliminated altogether by infection with 858. But focus on this one. This particular bacteria which incorporated S1 and S2 spacer sequences was resistant to this phage. Why? Because it had these S1 and S2 in the CRISPR-1 locus. Similarly, another spacer sequence S3 from the same phage 858 made these bacteria also resistant to phage 858. Now focus on this, or maybe this one, wild type which had a spacer sequence from 52972 S4. This spacer sequence was from another phage. So when you plated this, infected this bacteria with this phage, the efficiency of plaque was very less. They were resistant because they contained a spacer sequence which allowed them to restrict the genome of this virus and did not allow infection to take place. Now you have certain bacteria which are cross-resistant to both of them. So in that case, what you expect to see is spacer sequences from both the phages in that bacteria. So focus on this one. A resistant bacteria which contained S9, 10, 11, 12, S9, 10, 11, 12 from both the phages. So that's why these bacteria are resistant to 858 and the second phage. So it really depends what kind of spacer sequences are there or have been acquired by the bacteria that makes it resistant to the invading phage. Clear? Any question? No questions? So all the new spacer sequences keep on adding to the leader sequences just after the leader sequence. So this is leader sequence, you have the addition of all these spacers. Depends, the resistance towards invading viruses is going to be provided by the spacer sequence that has been acquired during the infection of the bacteria with the corresponding phage. If there is no acquisition of this particular DNA sequence from the invading phage, that bacteria is not going to be resistant. As simple as that. So this is how this was discovered, that something is there which is acting as an adaptive immune system for the bacteria. So the gene editing part, now this was actually exploited by the scientists later on to make it as a tool for gene editing. Okay. After understanding of how this actually is being used to restrict, this is just till now what we have learned is that you need spacer sequences. But how these spacer sequences are being utilized to restrict the invading pathogen DNA. After the understanding of that, it was actually utilized as a gene editing tool. So this is how CRISPR-Cas9 works. We are going to see how it was working to restrict the DNA, and then later on how it was actually adapted as a very efficient gene editing tool. But before that, how it works is, the present CRISPR-Cas9 system, how it works is, this is the target DNA sequence. You make your guide RNA, which is complementary to the target sequence. Okay, that is depending on what kind of Cas you are using. So different kind of guides can be designed depending on the Cas. It's not only Cas9, there are different Cas, 12, 13 and all so and so forth. So this is recognized by the guide sequence, and then adjacent to the PAM motif, proto-adjacent motif, in which in case of Cas9 is NGG. N can be any nucleotide and has to be followed by GG. So when you have that particular sequence recognized through this guide, you have in the same RNA that you make nowadays, you have the guide appended to tracer RNA. So you have a complete sgRNA, single guide RNA, that incorporates the guide and the tracer RNA. You make it as a single component that actually guides the Cas. Cas is again the nucleus. So two components, just remember those two components. One is for recognizing, the recognition part is being provided by the sgRNA here. The RNA molecule only is providing the recognition part, and the restriction part is being imparted by Cas. Clear? Okay. So this is the paper that actually showed the functionality of CRISPR. This is from the Jennifer Doudna's group. Okay, so you have this programmable dual RNA guided DNA nuclease in adaptive bacterial immunity. So what I told you in the last slide was, you have some spacer sequences acquired. But how these spacer sequences are working? So the Cas proteins that you saw, those Cas proteins actually process the RNA molecules that are produced through these spacer sequences. So two different kind of RNAs are produced by these CRISPR-1 locus that we saw in Streptococcus thermophilus. One is the CRISPR RNA, the CR-RNA. Other one is tracer RNA. So you have this, the CRISPR RNA is the one that contains that spacer sequence. Okay, so you have these two RNA molecules together, CRISPR RNA, tracer RNA. There is some sequence which allows them to complementarily bind to each other. One of this, actually the leader part, is having a complementary sequence to the target DNA. So what are the different components? One is CRISPR RNA, other one is tracer RNA. There has to be some complementarity to allow them to join together, the CRISPR RNA, tracer RNA. And this combination of CRISPR RNA and tracer RNA should be able to recognize the target sequence. And that sequence is present in the CRISPR RNA. Clear? This will allow it to complementary bind to the target sequence. So this is the complementary strand. This is non-complementary strand. So near the PAM motif. PAM motif is NGG. Clear? So it is going to bind here, and then after it has bound, then the restriction, the nuclease, the Cas nuclease, endonuclease is going to restrict this, introduce the double strand break. So this is how the system is working in the bacteria. Okay, so now focus on this, how they actually deciphered all this. So you need to have all the components present, starting from the Cas9. So if you do not have any of these components in the reaction mixture, you have the target sequence. You provide the target sequence in vitro. This is all in vitro reaction they proved. So you see all these lanes. Just focus on this lane first. Why you see this 23 nucleotide fragment here? Because the target DNA must have been restricted. Okay, must have been fragmented. So why it has been fragmented? Because it has the tracer RNA, it has the CRISPR RNA, the corresponding CRISPR RNA, SP2. If you have any other CRISPR RNA, SP1, which is non-complementary to the target sequence, you will not be able to restrict the target DNA. Here, this one. If you have SP1, no fragmentation, right? Only when you have SP2, then you see this 23 nucleotide. So you should have tracer RNA, you should have CRISPR RNA SP2, you should have Cas9, and you should have the strand. Then only you will be able to see this restricted fragment. If any of these components is missing, you will not see this fragment. Now you can see this. Why you do not see anything here? Because nothing is there. Why you don't see here? Because only the strand was there. Why you don't see here? Because Cas9 was there, but other components were not there. Why you don't see here? Because SP2 only was there along with Cas9, but there was no tracer RNA. So how the endonuclease is going to, because endonuclease for its pairing with the CRISPR and the tracer, it needs to have some sequences in the tracer RNA. So if tracer is not there, then also you do not see this. If SP2 is not there, then also you do not see. Clear? So you need to have all these components together. But to make it more user friendly, the same paper, what they did was, they combined these two, CRISPR RNA and tracer RNA, into a single RNA. So why would somebody make separately CRISPR RNA, tracer RNA, two different expression systems? Why do you need to do that? So what they did was, they combined together this into a single RNA, that is sgRNA, where you just need to keep on changing your guide sequence, this sequence. You keep changing your sequence, the guide sequence, tracer is the same. Change the guide, retarget this particular CRISPR to any locus in the genome. That's why this is so popular. Okay? Any question on this? Any question? I hope you understood this functionality of this and how it was actually deciphered as the adaptive immune system first in bacteria, how it works in bacteria, what is the recognition mechanism, and then how it was actually re-engineered into a system which can be used as a gene editing tool. So now in our lab, we use a lot of this genome editing tool, CRISPR-Cas9 specifically. what we do is we manipulate the genome of the malaria parasite. So malaria parasite, there are different malaria parasite species that infect humans. So what we work on is Plasmodium falciparum, the most lethal species of Plasmodium that infects humans. So we also do a lot of gene editing using CRISPR, be it nucleotide substitution, introducing SNPs, indels, deleting something, adding something, tagging the endogenous locus with these epitope tags, knocking out, conditional knockouts, everything we do. So what we do is we actually started with 2 plasmid system. So you need to express all the components in the CRISPR. What are those components? Cas9 is there, then your guide sequence is there along with the, so now we consider guide as a single sgRNA. Okay, so earlier the way it was, this evolved in the malaria field was that you have a 2 plasmid system. You will express Cas9 from one plasmid and then you will express your guide from another plasmid. To have a user-defined manipulation or modification of the target gene, what you need to have is, you need to also provide. So with this system, you will only be able to restrict the target sequence, the target site. But to incorporate your desired modification, what do you need to have? Third component. Template. Not template. Yes. You need to have a repair template, repair sequence. If you do not have that, then what will happen? Will you be able to incorporate your desired modification? No. Because what I said was 2 pathways, NHEJ and HDR. So if your purpose is to incorporate your desired modification, then you need to provide a repair template. And that repair template is going to contain all the modifications that you desire to incorporate. Okay, so here if you focus, let's say I want to modify this locus, CDPK1. Okay, so what I need to have? I need to have a plasmid that will express Cas9. This is going to express the Cas9. I need to have a second plasmid which will express the sgRNA. Why sgRNA? Because I need to have a sequence, the sgRNA sequence, which is specifically target my Cas9 to CDPK1 locus. Clear? And in addition to that, I need to provide a repair template. And that repair template is here, right here, with the modification that I want to incorporate. So suppose in this whole sequence, I want to change a nucleotide here to something else. I wanted to do some specific mutation. I told if we can check the function of a gene under in vitro conditions, then we want to ask a question, what will be the repercussion of this change in functionality on the overall phenotype of the microorganism? So this is what I intend to do. So I want to see what will be the change of this particular nucleotide on the overall phenotype of the microorganism. So I restricted this part. This system now, after introduction of the double strand break, is going to use this as a template for repair. In the process of the repair, homology directed repair, this is going to be modified. So now at the end of this whole process, I will have a parasite line or strain which will have CDPK1 with the desired modification. Clear? Now I can ask a question, what are the consequences of this change in the sequence of CDPK1? Similarly, I can do knockouts. Like for instance, what will I do in that case? I will have a sequence corresponding to this HDHFR. HDHFR is actually a positive selection marker, human dihydrofolate reductase. So, and another HDR corresponding to this, and then these are going to double homologous recombination, and then this is going to substitute the fragment in between. So the whole gene is going to get deleted. I can knock out things. Okay, so after this, what you are going to see is something like this. So this is your target sequence in the CTPK1 sequence. You have your modified sequence, this. You want to change ACC to ATG. This is the desired modification, ATG. You want to change, I think this one is threonine to methionine. ATG is methionine. So threonine to methionine. So you have your modified DNA provided as this, and then after the sequencing, then what you will have is T is substituted to M. How will you confirm this? If you want to confirm whether your desired modification has been incorporated, how will you confirm this? PCR? PCR is a little bit tricky because you need to have sufficient, mismatch in the sequence of the two, the wild type and the mutated one. If you have incorporated sufficient mutations here, like in this, it can be used, but again, depends on how much mismatch you have in the repair template. One thing that will definitely going to answer confirm is DNA sequencing. What you do is go back, you amplify this whole segment from here to here. Okay, you amplify the locus in the modified parasites, and if those modified parasites contain your desired modification, you will end up seeing this in the DNA sequence. That's the way to do it. Clear? PCR can also be used, but the only thing is that you should have sufficient mismatch somewhere in the primer binding sites. Like here, if you see, there are quite a few mismatches, and these are intentionally introduced. There is a reason to it. Can you think of any reason? Anyone? Why do you see so many mismatches here? And also a word is written, shield mutations. What comes to your mind when you hear this shield mutation? Something is shielding. So these are shield mutations because they are going to protect your modified DNA from recutting by Cas9. Because if you do not modify, then that particular modified locus will be recognized by the same system again and again and will keep cutting it. So to avoid that, what we do is we change this particular sequence, the guide sequence. So if you change the guide sequence, what's going to happen? It's not going to recognize, it's not going to take the Cas9 to that CDPK1 locus ever again. Understood? Any confusion? Any question? You can ask, feel free to ask. No? Okay. So these are, some of the results from my published studies. Similarly, like CDPK1, I showed you, we also knocked out CDPK2. This is how you do. This is what I was describing to you, that you clone these fragments corresponding to the 5' and the 3' regions of the target gene, flanking this HDHFR. But you restrict somewhere in between this locus. You introduce the double strand break somewhere in the middle, so that repair template can introduce this particular cassette into this. So after this desired modification, what you end up getting is this particular locus has been changed to this. HDHFR has been introduced in the gene sequence. So when you have something introduced into the gene sequence, obviously it's going to disrupt the function of that gene, because you will not have a functional protein made anyway. So I'll not describe everything, but how can you then ensure that you have modified the DNA, in this case in the knockouts? By PCR. Now you can use PCR to ensure that you have modified the target genome, target site, right? So you can use these primer sets. So F3, R1, R2, R3. So like F3 is common here and here also, R3 is common here also, but there will be a size difference. You will see a size difference because in the modified you have incorporated the HDHFR cassette. So you can use PCR for diagnostics to making sure that you have modified the DNA. So in addition to that, maybe I'll just wind up here. I don't know how much. I'll not go through all this. Maybe I'll just tell you about this. So in addition to that, what we have recently done is we have made our own indigenous strain whereby we can regulate conditionally the knockout of the gene. So you understand by complete knockout and conditional knockouts? What is complete knockout? Like the one that I showed you, CDPK2. Complete knockout means from the very beginning the gene is not present. What is conditional knockout? Yes, so at a specific time you can knock out the gene, not from the very beginning. What is the advantage of having conditional system versus complete? Especially in case of the study of essential genes. If your gene has an essential function, okay, then will you be able to generate knockouts, complete knockout? No, because why? You will not get that particular microorganism. Because if you completely knock out the gene, you will not end up getting the parasite. Because that is essential for the parasite. And if something is essential for the parasite, it will not survive, it will die. You will never get a viable parasite. But if you use a conditional system, you can get the parasite and then you can disrupt the gene at your own will. So there are various platforms that are used for conditionally knocking out genes. What we have used is a dimerizable Cre recombinase system. I don't know how many of you know about dimerizable Cre recombinase system. So the way this system works is you have two different fragments of a Cre recombinase. Cre recombinase actually recognizes certain small sequences, DNA sequences. When it recognizes two sequences, either arranged head to head or head to tail, it can recombine these sequences together and then lead into either inversion of the gene or knocking out of the fragment between these two sites. So suppose if you have something like this, you have this, then you have this. So this is LOXP. So what the Cre recombinase does is it recognizes these. The way these are oriented, if they are oriented like this, head to tail, this recombinase is going to recognize these, allow recombination to take place. This will loop out like this, and then what you end up getting is this intervening fragment is going to be deleted. So this is how it works. Now how do we make it conditional is you append something here in the recombinase, you split this recombinase into two fragments. So by themselves any of these fragments are not functional. Only when they come together they reconstitute the functional enzyme. And that coming together is regulated by a small molecule called rapamycin. When you have rapamycin, so that rapamycin has a binding site for both the fragments. So it allows them to be reconstituted. And once this is reconstituted, it is going to act on the LOXP sequences. So you have made the system conditional. So this is what we have done. We have actually used this particular cassette which expresses these two individual fragments. These individual fragments have been integrated in one of the locus called CG6 locus. Okay, so you should have a parasite whereby you have this recombinase expressed somewhere in the genome. Then to regulate the expression of the target gene conditionally, you need to integrate these LOXP sites into the target gene. Suppose you want to disrupt this particular gene conditionally. So what you will do is, this is the endogenous locus. This is how it looks. To make it conditionally regulatable through dicry, what you do is you manipulate your gene in such a way that you introduce one LOXP site here, another LOXP site here through CRISPR-Cas9 in the background of the parasite which expresses the recombinase. And when you add rapamycin to this, what will happen is, this is going to reconstitute this will act on this, conditionally you delete the gene. So two requirements. One, you should use a parasite strain which expresses recombinase, Cre-recombinase. Another one is, you should be able to integrate LOXP sites, two LOXP sites in the target gene. So we have been using this system now to understand the function of different genes, to identify essential genes in the parasite. Suppose I know that this gene might be essential. So what I do is, I take two LOXP sites, integrate them through CRISPR, put rapamycin and ask a question, what is the function of the gene? Simple. So for this, we have made our own parasite strain that will express in first place the recombinase. Clear? So any question on any part that you have not understood, or otherwise if you want to ask something, discuss something, you can ask.
So any limitation of CRISPR-Cas9?
The limitation, I would say, is the off-target effect. Yes. But now, this system is being refined, better refined to increase the fidelity of the Cas9. So if you have a Cas9 which is very strict in its recognition of the target sequence, because some mismatches still this Cas9 can bear. Like the sequence that I showed, the guide sequence, it's a 20 nucleotide sequence for Cas9. But still, if you have some mismatches, especially towards the 5' end of the sequence, so you have these 20 nucleotides. Yes, it's there. Yes. So this is the guide. You see this? This is the guide followed by the PAM, G, 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 G. Now, if you have some mismatches over here, up till 12 nucleotides, that sequence still can be recognized by Cas. So what is the disadvantage of that? That it is going to even restrict the target sites which are not completely complementary to your target sequence. But now, with more advent and discovery, this is being made into more, the fidelity of the enzyme is increasing. So you will only recognize a sequence which is highly specific to the target sequence. The mismatches here towards the PAM motif are still not bared by the Cas, but still one or two can still be there. So this will lead into off-target effects. That's the limitation. Okay. Any other question? Very nice question. Any other question? Any question comes to your mind?
Sir, after random mutagenesis, how can you trace back the gene which is mutated?
After random mutagenesis, right. So the random mutagenesis, the disadvantage is that you will not have a control over where you are manipulating the gene, right? The genome. So the only way to know what part of the genome has been manipulated or changed in sequence, you'll have to get the whole genome sequenced. When you get the whole genome sequence, then you will come to know what sequences have been modified in my target organism. And you compare it to the reference genome. That is how you can. But you can instantly recognize what are the phenotypic changes in the microorganism. Suppose you grow a bacteria on some different medium which doesn't contain glucose. Let's say it contains some glycerol, let's say it contains. Then at some point, you mutate and then you change the media. From glucose, you substitute to glycerol. You will see that some of the bacteria, because of this random mutagenesis process, will start utilizing this as a carbon source something must have changed because of this random mutagenesis. You take that bacteria, isolate the genome, send it for sequencing. Okay? Any other question?
So can we detect or recognize the off-target effects
Yes, we can detect that. Again, the tool that I would use is the whole genome, or nowadays there are so many softwares that already predict that these are the likely off targets sequence information, because you have this 20 nucleotide sequence. So that software sees where are the substitution. So out of these 20 nucleotides, where all there is one substitution, where all there are two substitution, what are the places in the genome where there are 3 mutations, 3 substitution. So you already know the likely off targets when you select a guide. So now you know, but again, to precisely know where off target effect has actually taken place, you have to go for the sequencing. Okay? Good questions. So should we wind up? Okay.